Hi everyone, I'm Leyda and I study the different biomedical applications of magnetic nanoparticles at the Rinaldi Lab. So today I'm going to be talking about how we have attempted to enhance T-cell labeling by complexing transfection agents, which are cationic molecules, to Ferucarbotran, a biocompatible magnetic nanoparticle tracer which has a negative coating on it. Now, it's important to enhance T-cell labeling because sensitivity during imaging increases as tracer uptake into the cell increases, so we can have better imaging. Now, the first thing we did was characterize these complexes. We did 3 different concentrations of 2 transfection agents, poly-L-lysine and protamine sulfate, in both water and complete media. Now, we then proceeded to do in vitro studies with primary T cells where we incubated the cells with the complexes and then proceeded to do a cell count. Now, our data shows that there's aggregation for both poly-L-lysine and protamine sulfate, but more severely for the protamine sulfate sample as shown in our DLS data that shows hydrodynamic diameter. So then in our DMS, we could see a change in initial susceptibility indicated by a lower point in the y-axis for the protamine sulfate samples, indicating that in addition to aggregation, there was severe precipitation for the protamine sulfate samples. Then our zeta potential data for poly-L-lysine show a more positive overall charge on the particle in media versus in water for poly-L-lysine. Then our MPI measurements show that there's a constant signal with an increase in concentration of transfection agents for both transfection agents. Now, our in vitro studies show that there is a decrease in viability for both transfection agents, but more severely for protamine sulfate. Then our MPI data shows that a spike in the protamine sulfate samples, but also an increase in signal for our poly-L-lysine samples. Now, in conclusion, our Ferucarbotran with poly-L-lysine complexes caused minor aggregation, didn't significantly decrease cell viability, and didn't really affect MPI signal, suggesting that optimization of complexation could improve T-cell labeling. But we can't conclude anything right now because about T-cell labeling efficiency because of a difficulty in removing free particles from the supernatant, interfering with accurate data as shown in our figure 6 over here with the MPI data. Now, what we'll be doing in the future is optimizing the Ferucarbotran and poly-L-lysine complex by varying concentrations and other mixing conditions, and then evaluate the effects on T cells. Thanks, everyone.